Hi everyone, my name is Ujwal. I'm a principal SA and lead for healthcare and life sciences machine learning here at AWS. And I'm going to be joined today by Alison P. Heath, who's the Director of Technology and Innovation at the Center for Data Driven Discovery in Biomedicine at the Children's Hospital of Philadelphia. And today we are going to talk about how machine learning and healthcare interoperability can help accelerate pediatric research. This talk is going to be divided into a set of components, starting from some introductory, research about how clinical interpretation of genomic data is really important. We'll go over case studies and some AWS services that support genomic sequencing and analysis as well as interoperability. Now, to kick things off, I wanted to just introduce the audience to this relevant study that was published in the JAMA Pediatric Journal. It talks about the effects of early whole genome sequencing based studies in a population of acutely ill infants that was suspected of genetic disease. A portion of the population got early genomic testing within 15 days of their getting admitted into the study. And a portion of the population did not do that, and they got delayed genomic testing done on that population, which was about 60 days after they enrolled in the study. The findings came out. They realized that the proportion of infants who actually received a care management change was much higher than on the population that received early genomic sequencing than the population that did not. So that denotes that when you intervene early with genomic testing, you get some intelligence and knowledge from the genomic data set that you can then utilize to devise more unique care plans to the individuals. Now, all this underscores that genomic is at the heart of precision medicine. It's really difficult to ignore how much information is embedded within your genetic profile, and not utilizing that information for clinical research is a huge mess. So as and when these studies come out, it underscores the importance of adopting clinical genomics in your regular workflows. The reducing cost of sequencing is also a big factor for the clinical institutions to think about developing their own labs and utilizing genomic sequencing as a part of their regular workflows. But there are some inherent challenges. Analyzing this at population scale produces a large amount of data sets that are difficult to interpret and operate on and catalog and maintain. It's also difficult to analyze that data using conventional techniques. So cloud computing is a way in which you can, apply machine learning algorithms and techniques at scale on these large data sets and derive some intelligence that can then help, physicians to accurately design a care profile and care for their patient in a unique manner that their genetic profile and their living conditions allow them to. There's also this aspect in the genomic analysis around the large-scale data generation. Depending on the type of genomic sequencing you're doing, going from genotyping that involves like analyzing a few genomic samples for variations, to large-scale arrays that involve like looking at larger tiles, to exome sequencing, and then finally into whole genome sequencing, your size increases proportionately. And it's only a matter of time when all of this will run out your on-prem capacity. So it's really important to take into account how can you like design systems that can scale with your needs. And as you scale out your genomic sequencing and analysis platform, you cannot like be held back by under-par infrastructure. So cloud computing allows for all of this data to be stored in an elastic manner. And then, the application of machine learning on these data sets allows clinicians to design therapies that are very unique to each individual. As and when the customers are thinking about new and innovative ways to apply machine learning and digital therapeutics into the area of genomic sequencing, there are a huge advent in which the customers are looking at improving patient care or reducing the cost of care and increasing the cost, care quality. To help customers accelerate this transformation, AWS launched something called AWS for Health in the summer this year. And it is a collection of offerings from AWS services to vetted partner-based offerings that allow customers to realize the true potential of their healthcare data, including the genomic data sets, and then be able to apply them in a meaningful manner for insights. As part of the AWS for Health portfolio, there is a dedicated set of offerings for genomics that are divided into 5 different categories, from data gathering from instruments to data, cataloging and secondary analysis of that data. Also aggregation and making sure that that data is available for downstream machine learning models for interpretation. And then finally into clinical translation of that data into reports. All of this is, again, a wide array of topics, but we are going to talk about the interpretation portion and the clinical translation portion in our talk today. So now that we understand the full gamut of offerings or collection of offerings or different headers under which they belong, it's important to define what clinical genomic means. It is, at a high level, finding a solution to the way in which information embedded within genomic datasets allows for more targeted treatments and individualized treatments and diagnosis by physicians. It is needed because it allows for physicians to be more accurate in the way they design their care profile for patients. It also allows for getting to the decisions faster and quicker with more information at hand. And this is all something that can be enabled by platforms that support this kind of analysis at scale. There are some inherent challenges in the adoption of clinical genomics, and starts from security and compliance. So healthcare and life sciences data is highly secure, and genomic data is no exception. It's really important for the systems that store this information to be highly secure and be able to be protected by any attacks or hacks that can happen in those systems. The data sets and the results need to be reproducible. So you have to create pipelines and that are templatized and can be reproduced when the need arises. It also requires it to be agile, in a sense that the amount of time it takes for you to derive insights from genomic data sets need to be cut short. As we showcase in the original slide around the study that was produced by JAMA, you saw that early intervention with genomic testing is really important. So being able to early intervene and then draw insights and draw meaningful results from those insights is really, really important and key. And then finally, data sharing and collaboration. All of this requires collaboration from not only within the organization, but external collaboration with the researchers. So it's really important to be able to, design a system that supports that kind of cross collaboration and joint working mechanisms. So with that, I'm going to invite Alison to talk about her use case and how the Children's Hospital of Philadelphia designed a scalable genomic analysis platform for pediatric research. Alison?
All right, thanks so much, Ujwal, for that great introduction. I'm Alison Heath. I'm the Director of Technology and Innovation at the Center for Data-Driven Discovery of Biomedicine at Children's Hospital of Philadelphia. And I think it's a really incredible time where we have spent the last decade really building a lot of these platforms for genomics in the research area. And as you heard in the introduction, really have a new opportunity in the clinical area to bring data in real time, intersect it with that large landscape of research data, and continue to scale and create impact for patients everywhere. And really, this is part of the center that I'm at, D3B at CHOP, where we're a CHOP center of emphasis. And the mission here is really to continue to transform the healthcare discovery ecosystem and provide innovative care for all children through collaborative, data-driven science. And genomics is really a very key use case for us that is really at the forefront of what's happening with a lot of data throughout the healthcare system. And really, by bringing this all together, our vision is to make sure we have breakthroughs for every child, every time, everywhere, as we move through this unique period in time. And in order to do this, we have a very unique team of subject matter experts that we brought together. So we are not just data folks. My team, is a group of software engineers, DevOps engineers, data analysts, and others, but we work very closely hand in hand with others, such as the clinical research unit, which is consenting patients, working with them to bring their data in, the biospecimens, so actually banking the biospecimens that are getting genomically sequenced, other different molecular assays, really working together with wet lab units in order to create molecular diagnostics, in order to create cell lines, other translational research models to do preclinical testing and other things. The bioinformatics unit, who uses a lot of the cloud-based platforms to do the really advanced analytics pipelines and others. The DAP unit, which is the unit I run, as well as all the coordination that it takes to really have a unified way of going from specimens to impact. And you can really see this in our research methodology. So we really think of, the patients, that analog space of patient care, taking specimens, tumor samples, blood samples, other things, genomically characterizing them, bringing that data together, which might, from a genomic standpoint, has become quicker and faster, but bringing in the clinical information, the other information associated with it, which might be in disparate places, harmonizing that, creating a base of information that we can then use different tools and other things to bring new knowledge, new discoveries in quicker and faster ways. So ultimately, the impact is brought back to the patient for better outcomes, better care. And this is especially critical in the area of pediatrics, where many of the diseases are relatively rare. There's common pediatric diseases, but, relative to the general population, we often need to be able to connect different cases across both the nation and internationally. And really, this has motivated a lot of our platform design, a lot of the technology choices we've made to really accelerate this process of samples to impact. And so I'll just talk about one key consortium that our center works on, and this is called the Children's Brain Tumor Network. And we bank the samples at CHOP, but it's a very wide consortium of multiple different institutes that are trying to collaborate and trying to address a lot of those challenges you heard from Ujwal about how do you do data sharing and collaboration across multiple institutions, not just within your own. And this challenge, there's no other critical pressing challenge that could be more key to this, especially as brain tumors are the leading cause of disease-related death in children in the U.S. and also numerous other countries around the world. And furthermore, it's been, there's never been a drug directly developed to treat pediatric brain tumors, and it's becoming a critical need. And as we understand and bring these different centers together across both, as I said, the nation and the world, we need platforms to be able to provide this data, provision it, create workspaces, allow people to collaborate in different ways, come up with new techniques, and really continue to drive that mission. And so the consortium has come together and has collected a very large number of samples across multiple different histologies. So just a little bit about brain tumors. There's multiple different types out there, multiple different histologies. Some of them are more common, some of them are more rare, but generally, again, related to the relative population, is fairly rare. And so we need to bring all of those together, get as much information and data so that we can understand the underpinning reasons for why these horrible diseases are caused. And so you can see that we have over 4,200 participants, but they're across many different histologies, and trying to understand commonalities and differences across these different participants and research projects and other things that are moving forward. And so again, similar to our center methodology, the consortium methodology is to collectively work together to enroll patients on a research protocol that connects with clinical data and biospecimens that are related to their journey. And this intersects with different clinical trial efforts as well, where we can more rapidly provision the data both on research purposes as well as for clinical treatments. And then really being able to generate, as you saw, this large-scale data set. So moving to whole genome sequence, we all have 3 billion base pairs in our genome. A fully characterized tumor can generate over a terabyte of data. Even having 1,000, 10,000 patients fully characterized starts creating big data problems that we have been solving using the AWS platform. And then moving from there and moving back into the analog, how do you take that data, that collaboration from the computational folks, take it back into the lab, do more different experiments and have that iterative process where you have data, you go back into the lab, you test it, you find out more information that feeds back into your analysis, and that's how you get to the real endpoint, which are new therapies, new ways of doing treatments that we really want to make this process much, much faster than it has been to date. And, just to talk about that a little bit, a lot of this work is hugely collaborative, and it just has to be. The traditional research model, it was often single institution based. You were collecting a small numbers of samples. There was maybe one or two research projects going on. And I think key to what we've really done in building the platform design is making sure that data is not siloed or unavailable to those who are doing this kind of work. And so the CBTN research model has really pushed on how do we do it in a collective consortium way. How do we collect every sample, every surgery that's coming through, being able to collect any tissue that's not needed for clinical treatment so that we can use this for research purposes. And then having many projects being run simultaneously over this data, looking at it from all different angles, bringing all kinds of different subject matter experts, researchers to bear to really move brain tumor diagnoses more forward in more rapid time. And I think that really, like I said, has informed our milestones along the way from the consortium launch to really, the part of work that me and my team do in terms of building the platforms. And really, I say my team, but it's a collective effort with a lot of other groups, different national endeavors as well, of how do you bring together a genomics platform that is, cloud-based, that we can connect to both commercial partners, academic partners. Really, it's not limited to a narrow swath of research, but really, how do we connect all of these things together to leverage the best of breed to push this forward? And so, in 2016, even very early on, we decided it had to be cloud-based. We had to have shared infrastructure. We had to have key commercial partners at the time, Seven Bridges, who we still work very closely with, in launching the Cavatica platform. This has gone through launching the Pediatric Brain Tumor Atlas. We have also been working as part of the Kids First Data Resource Center to bring in genomic clinical data intersecting with other diseases, so not just brain tumors, but other pediatric cancers, structural growth defects, genetic disorders like you heard about the paper earlier, because a lot of times there are unexpected connections in the developmental space that are happening. It's not just about one organ or one disease. It's really about how do we find these different genetic variations that in one disease might cause something to turn off, and in another disease might cause something to turn on, but underlying it is a similar genetic profile. And then, as you heard, we are also moving to this mode, intersecting with clinical trial consortiums of we have this research landscape, we've collected a lot of genomic data. Now, every new patient that comes in through a clinical trial that can get sequenced, that data can be consented, shared with the research consortium, as well as informed on where on that research landscape does that new patient come in. And so if we go back to that methodology slide, you can start seeing some of the annotations here of how do the patient specimens come in. And you can see these key parts of, you have this initial platform, so the platforms that D3B have been building, as well as some of what we've been doing in the NIH space with the Kids First Data Resource Center. But importantly, how do you even connect to other platforms there? One platform, and here what I mean is a platform in terms of like a portal or a set of analytic tools aren't going to solve all the problems. Someone is going to be doing something maybe more specific for brain tumors. Other people might be doing something more specific for congenital defects. But you want to be able to connect your platform to other platforms, other visualizations, other tools. And you can only really do that through standard services, APIs, and interoperability. And so as we've moved over the last 5 to 6 years of going from the initial stand-up onto AWS to connecting to these other platforms, thinking about those APIs, thinking about how we connect them, that's also informed how we built out our architecture. And so we talk a lot about data to impact at scale. So how do you go from that left-hand side of that funnel to the right-hand side? And what are the different tools, architectures, decisions that we make, new services that we can use to really leverage? And as I mentioned, I think this has created a set of design principles where we really want to maximize the number of users and processes. We want a diversity of information, so we have many different data types that we're bringing in. Genomics and clinical data was a start, but we're also starting to bring in imaging, proteomics, other types of information. How do you integrate it? Then how do you have it connected? And then how do you have it flow rapidly in a secure way? You still need to be sharing with the right people at the right time, but to be able to have that provisioned and provided much more seamlessly than in the past, where you had to wait for large data transfers, or you did this in a very batchy way, once a quarter or something like that. We really want to think of things more on the week scale, day scale, hour scale, even minutes ultimately. And so over the last 5 years, we've really built out a lot of resources that have created over 1.4 petabytes of data on the genomic side, over 20,000 participants now. We also collect family information in pediatrics. You might imagine it's very important to understand the mother and father, genetics, and how that informs what's happening with the disease in the child. Over 26,000 samples, and really also characterizing the phenotypes of these. So not just the genomics, but pairing the phenotypic data with the genomics. And so if you think about the data integration problem here, we have multiple different data types, like I mentioned, and they need to come in, they need to have their own workflows for transforming them from data to information, doing that harmonization, if it's going to the research resources, de-identification, and then being able to bring those and integrate them for multiple different use cases. So just two that we've highlighted here is looking at integrated data for molecular tumor boards, where a new patient can be put on a backdrop of that research data landscape that I mentioned, as well as rapidly funneling the same information into the research and discovery pipelines that people have set up, that every new patient that comes in increases the power and understanding that we can bring to bear. So if we take that kind of the research, scientific, clinical use case and start looking at some of the architecture choices that we made over time, we have really tried to move to much more of a serverless, event-driven architecture. We make heavy use of AWS Fargate to have the right footprint at the right time for our different microservices that we use there. We have been increasingly looking at FHIR as a technology for healthcare interoperability, have been working with HealthLake and other resources in order to understand the different pipelines that we can do there. And then, really our kind of only footprint is EMR clusters, which allow us to do this large-scale variant cluster processing on the fly. We also use Elasticsearch pretty heavily for doing a lot of the searchability. So how do you find the data? How do you do aggregations? And how do you present and power some of the snappy UIs that some of our partners have been developing? And as you can see, we also link with other platforms. So this is our architecture. We've been working with commercial platforms such as Flywheel on some of the imaging data, Seven Bridges and Convatica for genomic data, and the University of Chicago's Gen3 platform for a lot of the security and compliance aspects. And because we all are in the AWS infrastructure, we can leverage different services. We can have smooth data flows, exchanges over S3 buckets, for example. So you can see in the middle there our genomic data and imaging data, which we stage. And now not only can our platform interact with that, but we can give access and use other tools right in line with it. So from here, I want to talk a little bit more about the genomic aspects and some of the challenges we addressed with the variant clusters over there on the right. And really, we needed to first create scalable cloud pipelines. So on the Convatica platform, we use common workflow language to allow our pipelines to be reproducible, Docker to package them up, and they're all available openly on GitHub for anyone to use. But we also have created an operations layer in terms of how do you track these, how do you understand the cost, how do you optimize the cost. Over the years, our cost per pipeline has dropped dramatically, and we've really been able to leverage things such as spot instances to be as quick and rapid as possible, while as low cost as well. And we're continuously monitoring this, making adjustments, optimizing the different instances and other things that we're using, and also improving as new bioinformatics tools come out so that we can do this in the most efficient way possible. The result of all those pipelines on the genomic side is very large-scale variant data. So for example, we have over 28 billion occurrences of variants across multiple different research subjects, and it creates a large data problem because researchers are used to using a database, maybe SQL queries, things like Jupyter notebooks, and really want to have a similar interface to this data, but you can't just do this on your local machine or even a very large EC2 instance, for example. And so we have been working together closely with partners at St. Justine's, which is a children's hospital in Montreal, who's developed techniques to map this data to Parquet. We store them in Amazon S3, and then we have Elastic MapReduce clusters over that are dynamically provisioned on the fly in using security rules based on the S3 buckets, as well as having Zeppelin notebooks as the ultimate interface that users are using to interact with this data. And this has been incredibly powerful. We can bring in other data sets, other public variant data sets. Again, many of these are already on AWS, so we can already use them, as well as part of some of the open data program there. And kind of going under the hood, this is the high-level version of it, but we can go down a layer. There's a lot of coordination that has to happen here. So, being able to provision these clusters on the fly, being able to check people's security and other things using IAM, using Glue as kind of a meta store to bring that into the different Spark jobs. And we suddenly start seeing that with this coordination, with the harmonization of the information and being able to stage this data for AWS services, we're starting to get to the point where there's enough data to start thinking about machine learning techniques, AI techniques that Ujwal will continue to talk a little bit more about, but really having that data already provisioned. And I think, some people would call it kind of AI-ready data sets to start doing some of this learning techniques. That is really where I think we're at a tipping point here with the different data sets as we've brought it into the different infrastructure and having that staged and ready. So if I just go back to that clinical genomics use case a little bit, the variant workbench, these other infrastructure, these large-scale data sets that researchers are using to understand better about these diseases is intersecting right with the clinical genomics coming in. And so how do we do this in more real time? And as an example, I was talking a little bit about this backdrop. So here we have a t-SNE plot of everything that was in the Pediatric Brain Tumor Atlas. And if you can kind of squint a little bit, you'll see there's a bunch of circular dots, and then there's a triangle kind of right in the middle. And that triangle is the most recent patient that this report is made for. So you have all the background of that research data, you have a new patient, you can do the rapid sequencing, immediately automate all those pipelines, and then be able to have this downstream analysis that does a t-SNE plot that puts that patient and says, what other patients are closest to it? How do I understand that? And then this can be put in the hands of clinicians through mechanisms such as the molecular tumor board, where they can go and look and start comparing and trying to come up with the best path forward for treatment of that new patient coming in. And this is just starting to happen, and we're just really on, I think, the tipping point of where this will go. And it's been really great to see all the different AWS services coming together to make this happen, and then really excited about the things you'll hear about next. Now that the data is staged, it's cleaned, it's ready to start doing some really exciting things, training models, doing predictive modeling, and understanding better, I think, intersections between data types, not just the genomics, which I spent a lot of time about, but also how do we integrate imaging, understanding of the clinical data. And so just to wrap up about this, looking back on the last 5 years of really why did we choose AWS and what has it supported, I think it's really accelerated the data flows from the data generation to the community availability. Once again, the past we were doing these things quarterly, even yearly sometimes. Now this happens in much more real time on the kind of the week to day scale. And I think we can even do better in the future. And utilizing just a really wide portfolio of services, going through those iterations, becoming more serverless, more event-driven with our architecture, rapidly scaling, maintaining the different views, and really bringing research and clinical processes together. And really, we're a fairly small team. Actually, we're really only two really core DevOps people with a lot of other people kind of pitching in part-time, but really empowering that small infrastructure team to be able to bring that data together. It's really critical, again, in the rare disease space to be able to bring every piece of information to bear on the treatment and also really not having to worry as much as we had in the past, kind of before we did a lot of the cloud-based platforms of security and compliance, leveraging the different services as well as other partnerships to do that, such as the ones I mentioned previously with Kavatica, Flywheel, and Gen3. So with that, I'll turn it back over to Ujwal, who will, I think, talk a lot more about some more exciting services and what you can do with all this amazing data on AWS. So thank you.
All right, thank you, Alison, for that incredible case study and presentation. I'm going to take you through the rest of this presentation and talk about how certain AWS services and features are helping out with clinical interpretation of genomic data sets. The application of machine learning has enabled genomic data sets to produce much more meaningful insights into how a care plan can be designed. And the application of machine learning to genomic data sets requires some specialized nuances that has to be taken care of when working with genomic data sets. The first service I'm going to bring up is SageMaker. SageMaker was launched a few years back from AWS and is now progressed to be a feature-complete end-to-end machine learning platform that allows researchers, data scientists, ML practitioners to design, develop, build, deploy machine learning models at scale. It comes with an integrated workbench, which we call the SageMaker Studio platform. The Studio platform allows researchers and data scientists to get an IDE experience for a variety of use cases that they can then walk end to end with. It allows them to interact with other SageMaker features and tools, all in a centralized manner. It also comes with a managed infrastructure built into it. So researchers and scientists do not have to worry about how to scale their algorithms, how to train in new and innovative ways, and then worry about infrastructure or whether it will match up to the infrastructure that they are designing for. So all of that is taken out of the equation. And all data scientists need to worry about is writing good code and developing good algorithms and machine learning models. It also comes with a lot of managed tooling that's built into it. Now, these are things that are totally optional. Data scientists can choose to use these tools for a variety of tasks that are pretty common in a machine learning lifecycle, or they can write their own or bring their own existing tooling into the platform. So in that way, the platform is highly modular. You can know more about SageMaker by going to that link. If I have to cover the whole of SageMaker today, it will probably be a pretty long time and won't fall into the bounds of this presentation. But I do want to bring up a few key features that SageMaker provides. And if you look at all of these features, they can be divided into multiple headers of preparing, building, training and tuning and deploying your machine learning models, which at a high level are the stages that data scientists typically go through when building a machine learning model in their own environments. So right from preparing your data, you would have tools that allow you to label that data at scale. This is provided by SageMaker Ground Truth. You can also prepare that data in a meaningful manner for machine learning using SageMaker Data Wrangler. You can use Feature Store to centralize all the features and run experiments from it, and then use SageMaker Clarify, which is a new launch of ours, to detect bias and unbalanced data sets before you send it through the training algorithm. On the build side, SageMaker provides pre-built Jupyter Notebooks environment that allows researchers to start iterating immediately. It allows you to run training in a variety of modes, either locally on the Jupyter Notebook or building models at scale on a cluster in a remote environment that you can connect your SageMaker environment to. You can also build automatic models using SageMaker Autopilot that runs through a variety of feature engineering steps, algorithms, and models to choose the best outcome for you. And then SageMaker Jumpstart that provides you a pre-built repository of solutions for very common and standard use cases. On the training and tuning side, SageMaker provides purpose-built open source list of containers that you can then utilize in your own algorithms. It also provides you pre-built algorithms that you can just provide data to. And it also provides you a way to train on distributed training libraries and manage Spot instances. Distributed training libraries are the ones that we are going to go a little deeper on today because it has high relevance to how genomic data sets can be utilized for machine learning interpretation. Finally, on the deployment side, there are different architectures and constructs, both from synchronous deployments to asynchronous deployments that SageMaker supports. You can use also the SageMaker Edge Manager to deploy your models on the edge device and synchronize it with the AWS cloud whenever there is network connectivity. And finally, SageMaker pipelines that allow you to create an ML ops pipeline for training your model, deploying your model, and then iterating through different experiments and updating your model in production. All of these features are available as part of SageMaker, the single platform, through the SageMaker Studio IDE. So now let's get a little bit deeper on the distributed training libraries that SageMaker provides. As you start training with higher volumes of data, especially in the case of genomics, as we were talking about, that the data growth is highly correlated to the amount of data you sequence, the number of people you sequence it with, and all of this produces a large repository of data that can then go into machine learning models to train. Now, the problem is that if you train on a single instance, you're having a bottleneck of the amount of time it takes for your models to train in a meaningful manner. Similarly, as the size of the data increases, the models need to keep up by designing more complicated architectures and complicated graphs that increase the model size, and it's really difficult to fit that model into the GPU memory for inferencing on it. So it's really difficult for both the data volume growth as well as the model complexity growth as you start thinking about designing your machine learning models with genomic data sets. Now, there are some inherent solutions available to counter this problem. On the data side, you can think about sharding your data and then parallelly loading it into multiple instances, and then drawing meaningful insights from each of those instances that can then be aggregated and synchronized across devices. On the model side, you can think about breaking the model up. If it cannot fit the GPU memory of a single instance, you can actually divide it up and fit the model into and distribute the model into other instances in parallel. There are some challenges to this. Now, in the data side, if you think about how the data can be distributed and the synchronization between various devices can happen, it's a highly complicated process. And some of these open source available distributed data platforms are not specialized for a certain implementation or a platform. They are mostly open source, and there's a lot of onus on the developer on how they maintain it, how they customize it for their own use cases. They also do not scale linearly. So as you increase the number of GPUs or nodes in your worker cluster, it becomes highly complicated to synchronize communications between them. And they introduce a lot of network overhead in this overall distributed training library. Similarly, on the modeling side, if you have to break your model down into various different smaller components, it's a really complicated process that requires a lot of understanding of the underlying model, its architecture, and sometimes it's not available or feasible for researchers to have a full understanding on how to break that model. It's also a very experimental and trial and error-based process that can take a lot of time. Also, when you break the model up into different smaller components and distribute it across a series of instances, to keep those instances completely utilized, you have to make sure that you're designing an efficient pipelining strategy that distributes the data in a manner that none of those instances are sequentially being operated, but they are being operated in parallel. These are some of the inherent challenges that come with the existing available solutions in this space. To solve this problem, SageMaker launched the SageMaker Distributed Training Libraries. There are two different training libraries that are available as part of the distributed training libraries on SageMaker. The first one is the Distributed Data Parallelism Library, and the second one is Distributed Model Parallelism Library. So the Distributed Data Parallelism Library shards the data, distributes it into different components on a cluster of GPUs. It also coordinates between those GPUs for efficient communication between them, and it's optimized for making sure that that communication does not introduce any larger overheads when you add more GPUs on the AWS platform. On the model parallelism side, the library basically understands the model architecture and breaks the model components down into different smaller components, and then it distributes it into different instances that can then execute the jobs, the training jobs, and the runs in parallel. It also makes sure that there is no underutilization of any of the instances in the cluster, and keeps all of the GPUs saturated at all times. This allows this entire thing can be adopted via a few lines of code changes to your existing training scripts, and it also supports existing ML frameworks that work on distributed training and data parallelism libraries, like the PyTorch data parallelism libraries, as well as the TensorFlow hot So all of those components that are inherently supported in the open source community can directly be brought into this framework and extended. So let's look at how the data parallelism library works. When you want to call a training job with the data parallelism library, you have to first import the data parallelism library into your training code. This can be done via the SageMaker Python SDK that the distributed data parallelism library is a part of. And once you import it, you are then able to call a training job on SageMaker with the data that's stored on S3 or any of the other storage platform like FSX or EFS, and then be able to call a training job on a managed cluster of instances. When you call this training job, SageMaker automatically looks at your data, it shards it and distributes it based on your configuration parameters and the number of instances you provide. It then executes the jobs in parallel instead of sequentially, and then communicates between the GPUs for making sure that the gradients are averaged in a right manner. At the end of the training cycle, you end up with a machine learning model on S3 that you can then deploy either for synchronous inference or asynchronous inference. Similarly, for model parallelism, the way to do this is to begin by importing the model parallelism library that also comes with the SageMaker Python SDK, and then provide that training parameters into your training code and call the training job. When you call the training job and you have the model parallelism enabled, SageMaker automatically spins up the underlying infrastructure based on your configuration. It analyzes your model, your network architecture, and your graphs. It then decides to intelligently partition your model into different components and then distributes it into the individual GPUs on your cluster. It then executes the training on each of those GPUs by dividing your data into micro batches, and then distributes the micro batches as a pipeline so that they are not sequential in nature, but they are being executed in parallel. This is where the boost in performance occurs. Instead of waiting for some of these processes that are inherently sequential to be executed in parallel, increases a lot of headway in terms of how much you have to wait for the training job to complete. At the end of it, again, you end up with a machine learning model that's trained as an artifact on S3 that you can then go and deploy as usual. So this performance benchmark slide shows some of the training throughputs as measured by samples per second for various models with and without the parallelism library enabled. You can look at the results, and it shows that SageMaker model parallelism library performs better for large NLP models with billions of parameters, like the T5 that has 3 billion parameters in most. And in most cases, these large models produce out-of-memory errors without model parallelism. So it's really, really difficult to, train such complicated and, highly parameterized models without scaling up the GPU. And even in those cases, it's not enough to fit it even in the largest GPUs. So model parallelism library actually allows for these kinds of models to train efficiently. So now that we understand how to apply ML on large-scale genomic data sets, I want to switch gears and go into the clinical interpretation with some of the clinical entity extraction from genomic reports. And even in literature where a lot of this is embedded. 80% of the patient care information is trapped in free-form medical texts. So these free-form texts include things like insurance claims, visit summaries, discharge notes, pathology reports, diagnostic imaging reports. There are so many different ways in which unstructured data is generated in the healthcare ecosystem. And in most cases, customers choose to either use rudimentary rules to extract information from this, which is not really accurate. And in other cases, they just like ignore it because, it's so difficult to gather all of this data, understand all of the, different connections that this data has to make with the genomic interpretation, for example. And then also, turn it into meaningful insights. It's really, really difficult. So AWS provides a service called Amazon Comprehend Medical. This is a named entity recognition service that recognizes different clinical entities from unstructured clinical notes. These entities are clinical entities are things like medications, medical conditions, tests, treatments, procedures, anatomy, and PHI. These are high-level entities with more granular representations as you go into each of these entities. The model also has the ability to extract relationships. So if you have, let's say, a test and a value associated to it, the model will automatically understand that and extract that information for you. If you have a medication and a dosage associated to it, it will automatically extract that too and relate it to the standard medication. So this allows for providers to get a more holistic picture of what their clinical note is talking about, or what information is embedded that might be clinically relevant in the report that they are looking at. Finally, the model understands traits. So traits are things that are not really a part of any entity, but adds a lot of value in terms of the holistic understanding of that entity. So things like negation can be a trait that can be caught by the Comprehend Medical models. Negation are things like the events that didn't happen. So like, let's say the note says the patient didn't follow the care advice or didn't take a certain type of medication or didn't have any signs of a certain complication. Those can be, caught by Comprehend Medical as a negation and attached to the entity in question. So you know that, this particular entity is being mentioned in the context of negation. It also understands temporality. So if you have like time events in your clinical notes, it will be able to extract that. And then, the other form of traits are diagnoses, signs or symptoms that can be then attached to the clinical conditions that the models detect. Comprehend Medical also has the ability for extracting ontologies and linking it to standard terminologies like ICD-10 and RxNorm. ICD-10 are really important in the context of standardizing your clinical conditions. It's used by a variety of downstream systems, like billing, for example, to find the right, charge codes for various clinical conditions or treatments that are mentioned in your care plan. RxNorm codes are great for like drugs name standardization and pharmacovigilance. So if you are looking for like brand names that can then go map into standard drug codes, you are able to then standardize them using RxNorm codes as one of the options. So with Comprehend Medical, there are dedicated APIs that allow you to send a payload and be able to extract all of the ICD-10 codes and RxNorm codes that are embedded in that text in a very easy manner. And these APIs, both for the entity detection as well as the RxNorm and ICD-10 detection, are available in the choice of your language through the AWS SDK. It's also available as a CLI, so a command line interpretation of this entire exercise can be done via like bash scripts if you want. And then it also works both synchronously and asynchronously. So if you have an architecture that expects you to get a request for, a response for an immediate request to a call to this API, you can do that synchronously, or you can orchestrate these as jobs that can run asynchronously on a repository of nodes, crawl through them, extract their entities, and at the end of the job, you'll have all the extracted entities centrally available for you to analyze. So there are some benefits to Comprehend Medical, like being able to quickly and accurately extract clinical entities from free-form clinical notes and reports. This is a highly manual process today and takes a lot of time. And by having Comprehend Medical, you can automate this entire process and get faster processing, up to 25% for small charts and 375x for rare diseases. The service is also HIPAA eligible, which means that it allows you to store, process, and transmit PHI information through its control plane. This is great because the service is also stateless and it doesn't have any record of any payload that you send to it. It's able to extract information and give you back the information. There's no trace of any of that payload being remaining in the control plane of Comprehend Medical. So that also is a great mechanism to make sure that you're not having any compromising information being sent to an external endpoint or service and still be secure from a HIPAA perspective. So now that we've seen both the application of ML on genomic data sets and then interpretation and extraction of clinical entities from clinical reports that may be associated with genomic testing, we can combine the two into a single workflow and create models on multimodal data sets. This is kind of what Allison was also hinting towards, where they are extending their platform to take into account imaging data or EHR data or data from a variety of other systems that exist, different modalities. By utilizing an architecture in SageMaker that is highly modular, you would be able to use tools like AWS Glue that allows you to convert your VCF files into Parquets and then feed it into a SageMaker feature store. You can also use the SageMaker Data Wrangler to read EHR data that might be sitting in an EHR system in a structured fashion, and then be able to extract and normalize those features and combine those features into a centralized feature store on which you can run experiments. So this kind of architecture is again very modular and extendable. Like tomorrow, if you have an imaging sort of workflow that you want to extract features from, you can feed them into the same architecture and centrally store its features in the SageMaker feature store as well. And all of this, allows you to run centralized training on SageMaker. So SageMaker experiments can kick off, hundreds or thousands of experiments in parallel and be able to use the same set of features. And then you can choose the best one that actually works for you in terms of accuracy or the metric you're tracking, and then deploy them on SageMaker for actual inference. So in the last part of this presentation, I'm going to talk about AWS's push into the space of interoperability and what is AWS doing in this space, specifically for customers who have already jumped on HL7 Fire as the choice of their data store and using HL7 Fire messages for analyzing and aggregating patient data. So it was announced last year that we've launched a service called Health Lake that was in preview, and then we recently went GA with Amazon Health Lake. Health Lake is a service that allows you to import data from a variety of healthcare systems that supports Fire interfaces. If you have data in non-Fire formats, you can easily convert them through some of the partners that have integrated offerings on top of Health Lake. And then once you import that data on Health Lake, you get a managed data store that you don't have to worry about scaling and maintaining and securing. You also have the ability to automatically transform that data using named entity recognition models. So anytime your healthcare records or Fire resources have any unstructured components into it as a document reference resource, for example, Health Lake would automatically extract clinical entities from those documents and then make those available for search and querying on top of that platform. So you're not only being able to query information that are structured within the confines of your EHR, but if you have clinical notes, you can query and search on them too. Finally, SageMaker and other analysis tools fully integrate with HealthLake. So if you have machine learning models that can benefit from extra set of features that are extracted automatically from HealthLake, you can feed that into machine learning models built on SageMaker and get a boost in performance. These are some of the studies that we have also published ourselves, like working with a variety of data sets, extracting extra bit of entities that are extracted by HealthLake that gives a boost in performance in the overall model versus just using the bare bones entities that were previously available without HealthLake. And I know we've talked about HealthLake and Comprehend Medical, which are the first two tiles on the slide, but we have a ton of other services that are available as well as under development that allows you to work with clinical and medical data. Like, for example, Amazon Transcribe Medical is a service that allows you to automatically convert clinical speech into text and has a lot of applications in the speech in the space of telemedicine, ambient listening, and pharmacovigilance and clinical trials as well. Then there is Amazon TextRack that allows you to extract raw text from PDFs and images, and also be able to integrate that with Comprehend Medical as a downstream tool, so that now you're building like a very intelligent document processing pipeline that goes and extract automatically the entities that are clinically relevant directly from the documents, instead of like storing them at blobs or large file formats that you don't really have easy access to query on. And then finally, Amazon Rekognition, which is an image recognition and image classification service from AWS. It works on a variety of images, but it does have the ability to train like a custom model using labels that you can provide, a set of labels and a set of sample images. So Rekognition can like utilize those labels and sample images to automatically create a version of the model that is relevant to your use case. And this can be done with medical data as well. So if you have images of cells, for example, that shows anomalies in certain cases and you have them labeled, or you have like radiology images that have bounding boxes or certain kind of labels associated to a particular clinical conditions that it denotes, you can feed that as a label into the Rekognition custom label and design like a machine learning model that is very specific for that use case. So with that, I want to conclude today's session by saying that this is just at the tip of iceberg. We are seeing a lot of interesting patterns emerge from the study of genomics and moving that into clinical genomics specifically, and utilizing all of that information into meaningful clinical research and meaningful therapies that can be deployed at the point of care. We do have a lot of other interesting talks in our track, going from a lot of machine learning oriented talks to interoperability related talks and so on. You can see all of them in this slide deck slide here, and also go check them out in the catalog and add them and attend them if you find these area and topics interesting. So thank you so much for listening in, and I hope you found our talk valuable. Hope to see you next time.